Thank you, Chris, for the kind introduction. Thank you to the organizers for giving me the opportunity to present the work here. I will be the first talking about epigenetics. DNA methylation is really the focus of my presentation, and I have a few very basic introductory slides, some published work, but then also I would quickly transition into unpublished work that I would like to share with you. Okay, so DNA methylation is basically the addition of a CH group to the cytosine, which creates the 5-methylcytosine. The enzymes are known, DNA methyltransferases, that confer this reaction. However, when you look at the epigenome of cells, DNA methylation is not the only modification. There are also histone tail modification. The positioning of the nucleosomes becomes important. However, the concept that has worked out by many groups in the field is that epigenetic patterns, epigenetic modifications regulate gene transcription. And it's not as simple as it's shown here on the slide, but basically you can view epigenetic modifications as regulator of gene transcription. And there will be many more talks by Iñaki later on talking about all the different sites, enhancers, and so on that become epigenetically regulated. So since we started with a bit of history, I think over 20 years ago, John Bird and I at the Ohio State University came together and discussed the possibility to scan genomes for aberrant DNA methylation. I just want to show you this. I don't explain this, but this was the first genome scan in CLL, looking at about 3,000 genes in the CLL genome. And we had some very interesting findings, which actually have to be now reinterpreted. And I will introduce you slowly to this. What happens is these RLGS patterns can be analyzed by overlaying profiles and looking for differences in the spots. This was the job, of course, of the graduate students that were very happy to do this work. Today, you would do this, of course, automated with computer softwares and so on. However, the technology was incredibly powerful in identifying genes that are methylated in the CLL genome. Now, today we understand that the epigenetic pattern is much more difficult to interpret and much more complex. For example, when you look at what you consider normal tissue, there are different cell types at different developmental stages. So this, of course, relates also to epigenetic patterns within the individual cells. So each cell, basically, each cell type carries a different epigenome, which is imprinted on the DNA methylation level. The importance for cancer is, the question is at what stage and in which cell type do you get the transformation and which epigenome is carried into the cancer or into the leukemia. Now, this is a concept where Chris started as a postdoc many years ago, developing this idea and testing this idea in CLL. And I will show you a few slides from his really seminal contribution. So the first question that he asked was, is it possible to develop the phylogenetic relationship between different cell types from the hematopoietic system using DNA methylation patterns? And you can see nicely here in the tree, there are the hematopoietic stem cells. We have here the myeloid cells. We have the T and K lymphocytes. And up here we have the B lymphocytes, the naive B cells, and the mature B cells. And you can see already phylogenetically, there's quite a distance between them, indicating that they are different on the DNA methylation level. Now, the question, of course, is where do CLLs arise? Are they arising from cells closer to the naive B cells or the mature B cells? And this was the question that we tried to address. Before I go into the data, I just want to show you where the field stands. We now know that genetic lesions occur in pre-leukemic samples. BRAF mutation, Notch mutations, we just heard about all the genetic differences. And I will now show you that the methylomes are derived from cells that are closer to the memory B cells and that resemble cells that transition through the germinal center reaction. So we are talking here about two different events that are required for the initiation of CLL. Now, what we did together with Ralf Küppers and Mark Seifert, we sorted very rare cell populations. And at that time, we had established a technology to really map on a single CpG level the methylation status in a genome using very few cells, 2,000, 5,000 cells, fragmentation-based whole genome bisulfite sequencing. And these are the different cell types that we used to map these. This is something that we can do at our center in Heidelberg. We have a big sequencing facility. These are unfortunately very expensive experiments, but the data is of incredible complexity and incredible quality. So the analysis showed very striking features. First of all, you can see that about 25% of sequences, each line here represents a single CpG, shows gain of methylation. Dark blue in this case means high methylation, white means low methylation. And we see sites that gain methylation through the development from a naive B cell all the way to mature memory B cell. But the majority of sites here, and these are the top 5% variable probes only shown here, lose DNA methylation. Now, what is important to note here is that these are unidirectional changes. Once a site becomes demethylated, it continues to become demethylated. This is a program that runs through here, or the gains. And this is resembled here nicely on the phylogenetic tree. There is a linear relationship in this. There's no branching and no ups or downs. It's mainly going from one end to the other. This was our first very intriguing observation. Here I just show you data on whole genome bisulfite sequencing. This is a notched gene locus. And you can see this is not at all sites that we see DNA methylation changes. We see them at very discrete regions highlighted here and shaded in gray. These are distinct regions. These are cis-regulatory regions, including enhancers filled with transcription factor binding sites, indicating to us that very distinct programs act here that are epigenetically regulated. And many of these enhancer sites are known, but in this data set, of course, there's a lot of things to still discover. Now, when we look at the methylomes of CLLs, this is data from 250 CLLs, we see mainly three groups. Here, what we consider the low programmed group, the high programmed group, and something in the between. This is very similar what the Barcelona group has described. But what you also see is we saw this pattern before. These are very similar sites that we saw also in the normal healthy B cells, right? So the question that we then asked was, is it possible to use this information and map back the CLL cells to the trajectory of the normal B cells? And this is what we did here. And you can see now when we go by group, these are the low programmed, the ones from the left side, the intermediate, and the high programmed CLLs. They are all originating from different sites, but close to the memory mature B cells. But this is not the full truth. The full truth is this picture. This is one of my favorite pictures. Every CLL has a different origin for the DNA methylation. It comes from a different cell at a different maturation stage, right? So now this puts the whole epigenetic story upside down, because now we lost our control samples. So how do we identify now what are true CLL-specific epigenetic changes and what are changes that are relating back to the B cell programming? And this is, I have a few more slides. In these sites are also methylation events that we have reported before we even knew about this. This is methylation on the ZAP70 promoter. A single CpG site in here can predict outcome. There's an absolutely tight correlation of the methylation to outcome of CLL. This is shown here with a high methylation, low methylation, high methylation performing much better. So Justina Verbinska, a former graduate student in my group, now addressed the question, is it possible to computationally determine the methylome of the cell of origin from which the methylome is arising? And what she found is that modeling linear regression models for each CpG site, it's possible to determine the methylation level that a certain cell should have, and to determine at what maturation stage the CLL or the cell was, which gave rise to the CLL. This is basically shown here. If you plot the change in methylation, and you can determine at what stage the CLL was arising. And this is what Justina did. We found that there are basically 4 groups, those that arise from CpGs that usually are programmed by CLL specific patterns, but they don't behave properly. They overshoot. They lose completely methylation at a stage far too early, or they don't lose methylation. So these are hypermethylation, hypomethylation events. But then we also have sites that normally don't change, and where we start to see hyper or hypomethylation events. There are different, this has different functional consequences how these DNA methylation changes occur. This is the true data. You can see the majority are the hypomethylation event. Very few here are the hypermethylation events. This is the real data, naive B cells, mature memory B cells, and here are the CLLs plotted, deviating from 0 here. Now, initially, when we started our work with John Bird many years ago, we thought CLL is one of the cell types or the cancers with the highest degree of epigenetic changes. Now, this has to be reinterpreted now by correcting for the correct control cells and the control methylome. If we do this, this is plotted here. We can divide each CpG into different classes and calculate what is a CLL-specific event. And it turns out that the numbers are now very low. For example, in class B, we have only 0.25%, or in class C, 1% of the CpGs in the genome that are altered in CLL. Now, this is shown here. We basically overestimated about tenfold in the past the contribution of epigenetic alterations to CLL. This is now corrected, and now we have access to data that will teach us more about what are mechanisms behind this, what drives epigenetic deregulation in the CLL genome. I'm sorry, this is for microRNAs, this is for mRNAs, so very similar. And there were quite a number of reports on microRNA, epigenetically deregulated microRNAs in the literature, including our studies, where we overestimated the contribution of epigenetic alteration to the regulation of microRNAs. Now, if we look at the corrected data, we see very few microRNAs. Here, for example, class A, we have only 5 microRNAs left. Now, where we see clearly differences in the CLL epigenome, similar here in the class C. Now, what are these microRNAs? And this is something that we are just starting to analyze. Was striking. Quite a number of the reported target genes for these microRNAs are epigenetic regulators, enzymes that participate in establishing, writing, reading epigenetic information. So we are sort of going back in a circle. John mentioned SETD2, which is also an epigenetic regulator. So epigenetics and genetics is really tightly linked in many processes. This is already highlighted here in this type of data. Now, this is a final slide. This data just came out a few weeks ago, and we are still struggling to understand what this is. But what we saw is when we look at the mRNAs that become deregulated or overexpressed, we saw genes that are usually active in lymphocytes or T lymphocytes processes. So as you can see here, for example, for TIGIT or CTLA4 gene, they become expressed on the surface of CLL cells. And this was validated in a collaboration with Martina Seifert's group, and pointing to probably a much stronger effect of the microenvironment that affects gene expression in different cell types. Okay, I think I'm almost at the end. I'm in time, Chris. So, conclusions. So, normal B cells undergo massive epigenetic reprogramming during germinal center reaction. I think I convinced you. This is data indicating that about 30% of the whole genome is changing on the epigenetic level. This is very much in line with what was published by the Blueprint Consortium. Iñaki Martin Zubero was heading this work, where they also showed drastic epigenetic alterations during normal B cell development. We used linear regression modeling, which allows to compute the mesylome of the cell from which the mesylome arises. The term cell of origin may be or could be used twice, one for the genetic, one for the epigenetic part. But and what is important, previous methylation analysis overestimated the epigenetically silent genes in CLL, and data needs to be carefully reevaluated now with new controls. So with this, I would like to thank the participants. I mentioned Chris Oak already. He is now at Ohio State University. And Justina Werbinska in my group. She is now a postdoc in Jan Korbel's group. We have contributions by a number of clinicians. I mentioned Thorsten Sens, but John Bird, Stefan Stilgenbauer were part of this, and Carsten Rippe, Daniel Mertens, and Martina Seifert, here she is, working with us since a few weeks already, so with a very exciting new direction. Rainer Klaus was part in the early studies of the CLL. So with this, I would like to thank, and I'm happy to address questions.
Okay, we have a couple minutes for questions. Perhaps I'll start with one. The finding with the T cell genes is very interesting. So T cells carry themselves a very distinct epigenetic signature from B cells. If you sort of look broadly, can you detect a T cell signature within the data beyond just a small number of genes?
Yeah, very good suggestion. We have not done this yet. I mean, the data is just too fresh, but this is, of course, something that we have in mind, right? I mean, now trying to understand what is the reason, what activates genes, T cell genes, and what is the similarity to normal T cells, right? I mean, I'm happy to discuss this in more detail. I don't have a solution yet.
Wonderful. Hello? Also, speakers, please, or people asking questions, introduce yourself and your organization. Thank you.
It's my pleasure. Thank you. I'm coming from Nanjing Medical University, and I have a question. A few days ago, the Nature released an article on histone and DNA methylation relation. And my question is, what do you think about histone modification and DNA methylation in CLL? Thank you.
I mean, this is a big topic. There is clearly a connection between histone tail modifications and DNA methylation. There are proteins that read the histone tail information that allow other protein complexes, reader complexes, including DNA methyltransferases, for example, DNMT3A, to be recruited to certain sites. The intragenic sequences that become heavily DNA methylated in active genes, for example. So, I mean, but I'm sure there are many more connections between DNA methylation and histone tail modifications.
We have time for one more question.
So is it fair to say, based on what you've told us and what Chris has published and what others have said, that we really shouldn't consider CLL divided into a memory cell and a naive cell, that clearly all CLL come from antigen-experienced cells? They may differ in the presence of V gene mutations, but the absence of V gene mutations doesn't indicate that it's a naive cell. Is that a fair thing to say?
Yeah, I think that's a fair thing to conclude. I don't know if I have stressed this enough, but the maturation stage from which a CLL is deriving tightly correlates with clinical outcome. Right? The further the maturation has occurred, the better the survival. I mean, I showed the ZAP70 data, but there are thousands of CpGs that would do exactly the same thing, right? But you're right. I mean, the origin of the epigenome, the methylome, is coming from cells closer to the mature memory B cells.
Okay, thanks. Sorry,
Yes.
Rosie, I think we have to move on.
Yes, so Rickard Rosenqvist, Karolinska Institutet up here. Thank you very much. I fully agree with you that we have to reinterpret the data as you have been showing. But you showed this, your favorite picture of the diversity of the methylation landscape in CLL. So on the other hand, in the end of the presentation, you presented a linear model where you can sort of track back. How do you fit this together on an individual patient? Because if it's heterogeneous methylation patterns, how can you do it on a single patient?
Yeah, I mean, you need the data from the normal trajectory, of course, and then using the softwares that we developed to sort of place the methylome that is still present in the CLLs, in the background, right? The methylome of the cell of origin is still present in the background. Even if there are few CLL-specific changes occurring, you are still dealing with 10,000s of CpGs that are unchanged and that allow us to map back the CLL epigenome to a certain maturation stage. And we can talk about this
It's very
more.
exciting. Thank you.
Okay, thank you very much, Dr. Plass.